Next is about DNA synthesis inhibitors, their definition, example, inhibition, and resistance. So, what are DNA synthesis inhibitors? DNA synthesis occurs at chromosomes which require substrate, primer, template, and enzymes. We know that. Different drugs were used in treating diseases by inhibiting DNA synthesis in different manners. Analogues of purine and pyrimidines, which are used in oncology, and fluoroquinolones, which are used in treating bacterial infections, that inhibit topoisomerase 2 and 4, and DNA gyrase enzyme that is used in DNA replication. The enzyme topoisomerase and gyrase have quite similar structure, but they operate differently. Gyrase remove stress, while topoisomerase plays a major role in removing knots that occur in bacterial chromosome. Gyrase removes stress, but topoisomerase remove knots. Okay. Subunits of gyrase are gyr A, GYRA, and gyr B, whereas subunits of topoisomerase in gram positive is GRLA and GRLB and PARC and PARB. These subunits contain different domains that are involved in DNA cleavage. Humans also share two types of topoisomerase enzymes that have the same type of amino acids, which is created an issue in generating quinols, quinoles, quinolones. Then DNA synthesis inhibitors, ciproflavin is shown, PABA, DHF, and THF, bacteria, ribosome, and DNA. Metronidazole, DNA topoisomerase, fluoroquinols, and quinolone. Then DNA synthesis inhibitors. Fluoroquinolones are the group of antibiotics that are used in treating infections caused by both gram-positive and gram-negative organisms. So, nalidixic acid, I'm not sure about the pronunciation. Nalidixic acid, which was introduced in 1962, was the first quinolone for clinical use. Based on their activity, quinolones are divided into different generations. They are bicyclic core structure, which include carboxyl acid group and carbonyl at position 3 and 4 respectively, which enhances the activity of quinolones. Quinolones are divided into three categories, which include pyrrolidinyle and piperzinyl and piperidinyl. Piperzinyl type shows wide activity against gram-negative organisms, while pyrrolidinyle and piperidinidyl shows wide activity against both types of bacteria. In 1977, gyrase was identified as the target for quinolones, and later it was confirmed that it was a primary target for quinolones in gram-positive bacteria. So, mechanism of action of DNA synthesis inhibitors. For cell survival, topoisomerases and gyrase generates breaks in chromosomes. These enzymes can fragment the genome. Quinolones increase the concentrations of these enzymes, which result in causing cell death. Quinolones convert gyrase and topoisomerase into toxins, so they are referred to as topoisomerase poison. In a non-covalent manner, quinolones bind in the cleavage ligation active site and interact with the protein, causing intercalation into the DNA. As a result of intercalation, the concentration of cleavage complex increase the block ligation. So quinolones are there. And you look at the structure. DNA polymerase complex is their application for quinolones, and then topoisomerase also there. So protein independent SOS DNA repair, protein dependent cell death will be happening. So quinolone antibiotics interfere with changes in DNA supercoiling by binding to topoisomerase 2 or topoisomerase 4. This leads to the formation of double-stranded DNA breaks that cell death in either a protein synthesis dependent or protein synthesis independent manner. Then after the collision of drug stabilized gyrase or topoisomerase with transcription complexes and replication forks, DNA cleavage complexes are changed to chromosomal breaks. Because of this DNA breaks, SOS response and DNA repair pathways are affected, causing cell death, as quinolones inhibit DNA ligation by stabilizing cleavage complexes. and also affect the function of both enzymes. So quinolones also act as catalytic inhibitors. Some major examples of DNA synthesizing inhibitors: ciprofloxacin, ciprofloxacin, levofloxacin, moxifloxacin, ofloxacin, methotrexate. Methotrexate interfere with DNA production. This stops cell growth and division, resulting in the slowing or stoppage of cell cancer growth. Since cancer cells in general divide faster and with less error correcting than healthy cells, cancer cells are no more sensitive to this damage. The primary action of methotrexate is inhibition of the enzyme dihydrofolate reductase, DHFR, the enzyme that reduces folic acid to tetrahydrofolic acid. The inhibition of DHFR by folate antagonists, methotrexate, results in a deficiency in the cellular pools of thymidylate and purines and thus a decrease in nucleic acid synthesis. Therefore, methotrexate interferes with DNA synthesis, repair, and cellular replication. Then fluorodeoxyuridinate. This is schematic diagram. Misincorporation into DNA, increased DUTP, DNA damage, deoxynucleotide pool imbalance, TS inhibition, increased TS expression, increased levels of DUMP. Fluorodeoxyuridylate, also known as FDUMP, 5-fluoro 2 deoxyuridylate, is a molecule formed in vivo from 5-fluorouracil and 5-fluorodeoxyuridine. FDUMP acts as a suicide inhibitor of thymidylate synthetase by inhibiting the deoxynucleotide biosynthesis. FDUMP stops the rapidly proliferation of fast-growing death cancer tumors, and it is therefore widely used as a cancer treatment. Then fluorouracil 5-FU performs as a substrate during part of the catalytic cycle, and it is only during the synthesis of thymine from uridine when it is combined with other molecules to form 5-FDUMP to produce an irreversible inhibitor of the thymidylate synthetase functions. This inhibitor leads to an imbalance of the nucleotide grouping, stopping DNA synthesis. Next, mechanism of resistance of DNA synthesis inhibitors. Mutations in enzymes may cause quinolone resistance. Genes that cause a chromosome mutation that codes for protein targets and plasmid-mediated genes. Mutation of serine or acidic residue decrease the affinity of drug and enzyme complex. And disruption of metal ion bridge may also cause quinolone resistance. Mutations at acidic residue decrease catalytic activity, while serine residue in gyrase and topoisomerase 5 does not affect catalytic activity. Then plasmid-mediated quinolone resistance. QNR genes that encode proteins and MCBG and MFA decrease the binding of enzymes to DNA. The quinolones were protected, and these genes bind to gyrase and topoisomerase and inhibit quinolones from entering into complexes. Then chromosome-mediated resistance, expression of porins facilitate drug efflux, which leads to resistance.